Welcome to this explainer. Think of this as your ultimate pro tutor masterclass for totally dominating O-level plant nutrition. If you're gearing up for your exams, you are in exactly the right place. We're going to bypass the dry textbook reading and jump straight into the precise concepts you need. I'll be clearing up those tricky mechanisms, and best of all, giving you some incredibly powerful exam-ready memory hacks to lock down those top marks. So let's transform those dense pages into a streamlined, high-yield visual journey. Ready? Let's go. Here is our quick roadmap for today's session. We've got 5 key checkpoints. First, the magic of photosynthesis. Second, proving the process. Third, rate and limiting factors. Fourth, the leaf factory. And finally, number 5, essential plant minerals. All right, section 1, the magic of photosynthesis. Let's dive right into this first block. We're starting with the absolute foundation of all plant life, which is how they actually manage to make their own food. Okay, so when it comes to the exact definition of photosynthesis, you gotta know that examiners are looking for very specific keywords. You literally can't just write plants make food from the sun. That won't cut it. You need to explicitly state that it's the process by which plants convert light energy into chemical energy to make carbohydrates like glucose, and they do this using raw materials, specifically carbon dioxide and water. If you hit those exact keywords on your exam paper, boom, you are securing full marks for that definition. Let's look at the actual equation that makes all this possible. You need 6 molecules of carbon dioxide and 6 molecules of water. But pay close attention to the arrows right there in the middle. See, the green pigment, chlorophyll, acts as this ultimate energy trap. It catches the sunlight and then uses that light energy to physically split the water molecules apart, which allows the hydrogen to join up with the carbon dioxide. And the final result? We get our target carbohydrate, glucose, plus a crucial waste product that you and I rely on to breathe, 6 molecules of oxygen. So once the plant actually makes all this glucose, what does it do with it? Well, here is a pro memory hack for your exams. Just remember the acronym SCRS. First, we have S for starch. Plants turn excess glucose into insoluble starch for long-term storage. Next is C for cellulose. Plants link those glucose molecules together to build really tough cell walls. Then R for respiration. The glucose is broken down in the mitochondria to provide immediate ATP energy. And finally, that second S is for sucrose, which you can just think of as shipping. Plants convert glucose into sucrose because it's soluble and absolutely perfect for transporting through the phloem to other parts of the plant. SCRS, memorize that, and you've got this entire subtopic in the back. Moving right along to section 2, proving the process. Now, examiners absolutely love testing your knowledge of practical procedures, so we really need to know how to prove in a laboratory setting that photosynthesis is actually taking place. Since starch is stored exactly where photosynthesis happens, testing a leaf for starch is basically our best indicator that the process just occurred. But you can't just drip some iodine on a fresh leaf and call it a day. You have to follow these steps. And please pay super close attention to step 2. It is a classic exam trap. First, you boil the leaf in water to kill the cells and break down the membranes. Second, you absolutely must boil the leaf in ethanol. Why? To strip out the green chlorophyll pigment. If you don't remove that green color, you will literally never be able to see the color change of the iodine later on. It'll just be a dark mess. After the ethanol, you soften it in water, and finally add your iodine tops. And this brilliantly illustrates exactly why we do this test. A variegated leaf is naturally patchy, right? Look over at the leaf on the left. It has green parts packed with chlorophyll and white or yellow parts that don't have any. Now, after running our starch test, look at the result on the right. Only the areas that were originally green turn that striking dark blue-black color. The white areas, they remain totally unaffected. This is undeniable visual proof that chlorophyll is absolutely mandatory for photosynthesis to happen. We can actually use that exact same logic to prove that light is required too. If we take a plant that's been kept in a dark cupboard for a while to drain all its starch, and then cover part of a leaf with a black paper stencil before putting it out in the sun, well, check out the result. After we do our starch test, the area that was covered up by the paper stays this orange-brown color. It couldn't receive any sunlight, so it couldn't photosynthesize. The exposed areas, on the other hand, successfully made starch and turned blue-black. Pretty cool, right? All right, section 3, rate and limiting factors. Now that we've proven how the factory actually works, let's put on our troubleshooting hats. What variables speed up or slow down this biological production line? To measure the actual rate of photosynthesis, we often use aquatic plants, like pondweed. Why? Because we can physically watch them producing oxygen bubbles. This classic experimental setup is just brilliant. We place the pondweed in a test tube connected to a syringe. As the plant photosynthesizes, oxygen gas travels up and is collected in the syringe. That allows us to easily measure the exact volume produced over a specific time limit, say 5 minutes. Also, notice that we add sodium hydrogen carbonate to the water. That's super important. It dissolves to provide carbon dioxide, ensuring that a lack of CO2 doesn't accidentally mess up our experiment while we're busy testing other things, like the distance of the light source. We can also track the gases using a hydrogen carbonate indicator. Now, this is a high-yield summary table, so you definitely want to commit this to memory. The indicator starts off red at normal everyday CO2 levels. If you put the pondweed in the light, it photosynthesizes way faster than it respires, sucking CO2 out of the water, and so the indicator turns purple. But if you stick that plant in the dark, photosynthesis shuts down entirely. It's only respiring, which actively adds CO2 back into the water, turning the indicator yellow. So how do specific factors actually affect the overall rate? Look at this curve showing carbon dioxide concentration. As you increase the carbon dioxide, the rate of photosynthesis just shoots right up. Which makes sense, it's a raw material, so the plant uses it to make more food. But notice how the curve eventually flattens out and just levels off? That tells us that at a certain point, dumping more CO2 in doesn't help at all. Something else in the environment has become the bottleneck. Temperature, however, gives us a very different, unique bell-shaped curve. As things warm up, the rate increases because the enzymes controlling photosynthesis are gaining kinetic energy. They're moving faster. But there is an absolutely crucial tipping point. If it gets too hot, past the plant's optimal temperature, those enzymes become denatured. Their shape gets destroyed. When that happens, the process breaks down entirely, and the rate of photosynthesis plummets drastically right back down to zero. Now, what's really interesting is how all these variables come together. This brings us to the core concept of limiting factors. A limiting factor is simply whatever resource is in the shortest supply. It's the thing holding the whole process back. The golden rule for reading these complex graphs in an exam is to look at exactly where the curve levels off. For example, on the lower green graph, increasing the light intensity stops increasing the rate pretty early on. Why? Because the CO2 is low, so CO2 is the limiting factor there. If we boost that CO2, as you can see in the blue curve, the rate climbs higher. Basically, whatever is actively restricting the plant's maximum potential is your limiting factor. Let's move into Section 4, the leaf factory. Let's look closely at the leaf itself and understand how its internal physical structure is perfectly optimized for gas exchange and light trapping. It really is a biological factory. Imagine we slice a leaf right in half and pop it under a microscope. At the very top, we have a waxy cuticle and the upper epidermis. These are thin and transparent to let sunlight shine right through them, like a window. Right below that is the palisade layer, where the cells are just crammed full of chloroplasts to catch all that light. Further down is the spongy mesophyll, which is full of air spaces so gases can diffuse easily. At the very bottom, we have stoma. These are tiny pores for carbon dioxide to enter, and they're guarded by specialized cells that open and close them. And slicing right through the middle of all this are the veins, the vascular bundles. They bring in fresh water via the xylem and carry away the manufactured sugars via the phloem. Okay, to easily remember the difference between those two middle layers, I want you to use this quick alliteration hack. Palisade starts with a P, so just remember, palisade is packed tightly with chloroplasts for photosynthesis. Spongy starts with an S, so remember, spongy has spaces for gas exchange. Palisade is packed, spongy has spaces. Honestly, that simple little trick will absolutely save you during a stressful exam. And finally, Section 5, essential plant minerals. Sunlight, CO2, and water are great, but plants also need specific minerals absorbed from the soil to truly thrive and survive. For your O-level syllabus, you really need to be able to provide a sharp, direct contrast between two main minerals, nitrates and magnesium. Plants need nitrate ions to build amino acids, which are then linked together into proteins for making enzymes and new cytoplasm. If a plant lacks nitrates, it literally can't build new cells, leading to severely stunted, weak growth. Magnesium, on the other hand, is the central building block for making chlorophyll itself. Without magnesium, there is zero green pigment, so a deficiency means the leaves will literally start turning yellow, usually from the bottom up. This diagram perfectly visualizes those exact deficiencies we just talked about. Look at the little plant on the far right, it lacks nitrogen, and as a result, it is drastically smaller than that big, healthy control plant on the far left. Its growth is completely stunted. Now look at the plant lacking magnesium in the middle. It might have grown a bit taller, but its leaves are pale and sickly yellow because it simply cannot synthesize chlorophyll. Farmers actually look for these exact visual cues in their fields to know exactly what kind of fertilizers they need to add to their crops. As we wrap up this masterclass, I want to leave you with a provocative thought that ties everything we've learned together. We saw on our graphs earlier that higher temperatures initially speed up photosynthesis, right? But we also saw that if it gets too hot, the plant's enzymes denature and the rate plummets to absolute zero. So, considering the rising temperatures of global climate change, what happens to the world's plant life? And by extension, our entire food supply, if the Earth's temperature pushes vast agricultural regions past their optimal peak? Think about how these tiny, microscopic cellular mechanisms actually dictate the future of our entire planet. It's massive. Anyway, thanks for joining this explainer. Keep reviewing these core concepts, practice those memory hacks, and good luck crushing your studying.